Yeah,
Maybe the next
luckily
couple weeks.
it's not too busy, but next week I'll be out because of testing, and I don't know about next week's schedule due to testing, but hey.
Yes. So for testing, I will be at a test site. So Mrs. Rittenhouse will not be here. We will have review lessons that you're going to be doing daily, but I will not be in class with you because I will be at a testing site myself. So it's going to get weird over the next couple weeks just because we have testing. I'll be at testing. A lot of you will be at testing. We're still kind of low on numbers here, so I'm waiting for everybody to come in. But again, every single one of you will have a test. You'll have the biology test for sure. Okay, I don't know if you have tests in other subjects, but you definitely have one test this year because you're in my class, right? And biology is a tested subject. So you will at least have a biology test. You have 4. Caden has 4. Justin has 4. Do others of you have 4? Algebra, geometry, history, ELA. A lot of us have 4. Okay. So you'll be out some days too. So just know if you are out testing that covers your hours for the day. All right. Don't feel like you need to come back and try to do on biology test day or on ELA test day that you need to come back and try to get your bio hours in. Okay. We are all out, and we're all out because that's what we're focused on for school for the next couple weeks is getting those tests in. Do your best. I'm at a test site, so I know. Be at your test site on time, be at your test site on the right day. We have these all scheduled out. So if you don't know what your schedule is, take today and go get a 2-week block calendar and write those times and dates down. Okay? Get those down because it's going to be helpful for you and your test site and it'll get your teachers back in your classroom sooner. Yeah, well, so they just a lot of times if you are still taking a test more than once, then what they'll do is they'll give you multiple attempts to keep getting higher scores. Oh, it's okay, Arya. You must have been sleeping good. That's a good sign. You need sleep, so that's all right. So yes, so if some of you are taking them again, that's okay. We're going to just really work to get as the best score you can on those because remember, we need a collective score of all those tests together. So we're going to just try to score as many points as we can on those tests. So if you didn't get a 5, right, we're going to give you more attempts to try to earn a higher score. Awesome. All right. So this week we are going over heredity. Okay. We're going to look at cells, DNA, how you inherit things from your parents. And yesterday we talked a little bit about a karyotype. Karyotypes, again, are a picture, a snapshot of the DNA from one of the cells in an organism. We talked about this is a human karyotype and that we know that because there are 23 pairs of chromosomes, right? There's 22 that are numbered, and then you have our gender determining pair right here. All right. So we talked about karyotypes yesterday. We're going to go into it a little bit more. We ended with crossing over. Crossing over happens in meiosis. Meiosis, and this is the way, remember, that our cells make what kind of cells? What kind of cell division are we making in meiosis? Yes, I like all these answers. Sex cells, gametes, haploid cells, right? Cells that have half the amount of chromosomes because they are ready to meet their other half, right? They're ready to meet their other half and become a whole cell or a diploid. Yeah, good job, Pip. So let's move on from there today and just practice a few more questions. So we've got a question here about crossing over. Would somebody want to read the question about crossing over? All right, go for it, Pip. We'll have you read this first one.
Alright, I might have to zoom in because sometimes, text can be hard to read. The diagram depicts the process of crossing over, which occurs between homologous chromosomes during gamete formation, and the graph shows the crossing over. What is the result of this process? A, an increase in offspring variation, B, the deletion of amino acids in proteins, C, an increase in mutations in the genetic material, or D, the production of gametes that are genetically identical. Thank you.
Perfect. Great job reading. Again, if we don't remember what a homologous chromosome is, and we're like, oh no, Mrs. Rittenhouse, I'm freaking out. I don't know what that means. We're going to cross out what we don't know. We don't know what logos means, but we might know that homo means what? Everybody type it. Yes, yes, Faith. Yes, Aria. Yes. Yep, Pip is telling me homo is same, hetero is different. Homo equals same. So we're looking and we're basically saying the same chromosomes. All right. So it's saying that the chromosomes that code for the same thing. So these chromosomes have the same spots for like eye color or hair color or nose shape or lip form. Okay. They switch because they have the exact same genetic information on them. They're the same code, same chromosome, and that's why they're able to cross over. So what do we think? What we talked about this yesterday, so I'm not going to explain. I want you to tell me in poll why do we need crossing over? What's the result of crossing over? Is it the increase of offspring variation? Is it the deletion of amino acids, the increase in mutations, and the production of gametes that are genetically identical? We're kind of all over the place in this one. So let's talk about one that is not the answer. The least chosen option right now is the deletion of amino acids and proteins, which I'm glad that we've realized this is a nonsense answer. Okay? Nonsense. We're like, that doesn't even make sense. We're not talking about proteins. Okay? We have an increase in mutations. I can see how maybe you're like, well, if they're switching stuff, maybe something gets mixed up. Seems logical. D, the production of gametes that are genetically identical. Well, if my chromosomes are switching information, I'm thinking that's going to be preventing genetic like the identical genetic combination. Does that make sense? So this is like, well, this is actually the opposite. So let's get rid of this one. Now readjust your numbers. Anybody else? Can you readjust your numbers? Or do I have to repull you? Repull. Let's repull now. What do we think? Is it there's just increased mutations? Or is it provide an increase of variation? Good. This is more convincing. Yes, it is the increase. Whoa. The increase of variation. Remember, that's why our chromosomes actually cross over in general, is to give us genetic variation. Okay, so that we aren't getting a whole slew of one traits from mom and a whole slew of another traits from dad. But because when we are making our sex cells, they're switching information. It's kind of like a Rubik's Cube. You know what a Rubik's Cube is? Does anyone know what a Rubik's Cube is? Those block cubes with all the colors? Yeah. It's like crossing over kind of does that. It mixes those up each time in different spots so that you have a lot of variety. All right? You have a lot of variety because all of the two alleles for all your DNA being swapped all over the place. When they do that and they make multiple reproductive cells, that's going to provide differentiation. Okay. Crossing over equals, why do we do it? For differentiation. So go ahead and stick that in your mind. Here's our process of meiosis. Remember, meiosis is really mitosis twice. Mitosis is why I can do this right now and I can't rub to my bone, right? Because my cells are reproducing clones of themselves. So mitosis, everybody put this in chat. Mitosis equals makes identical cells. Oops, I spelled cells wrong. That's all right. Mitosis makes identical cells. There are 60 of you in this room, so I want to see lots of that. Mitosis equals identical cells. So mitosis, we're making all the same Mrs. Rittenhouse cells over and over and over again. All right, this is for maintenance and repair. Just like if you get a cut, that's why you get another skin cell that looks just like your other skin cells, right? Because of mitosis. How do we get our sex cells? Well, it's meiosis. It's mitosis crossing over, mitosis again, right? Crossing over happens during metaphase when they meet in the middle. All right. If you don't remember, this is an easy, fun one to draw. I encourage you to go back and draw these for review. Okay, you start with one cell, the DNA replicates, the cell, they meet in the middle, they pull to the sides, you end up with two. And then those two cells go into division and they divide. So at the end of meiosis, how you make your sex cells, your haploid cells, your gamete cells, you start with one and you end with four. In meiosis, it's like mitosis happens twice. You see what I mean? It's one cell and it gets divided to two. And then we take those two cells and they each divide, right? They each get halved and then we end up with four. So it kind of runs that process twice. We have prophase, metaphase, anaphase, telophase. Cytokinesis is when they pinch apart and become two. And now we have two cells, and they each go through what we say prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2, right? Perfect. So that is the difference between mitosis and meiosis. One is mitosis is our somatic cells, our body cells. Meiosis is making our reproductive cells, and it goes through kind of that mitosis process twice, all those same steps. So let's do another question. Anybody want to read? I know Pip will read for us. Does anybody else want Ireland? Let's have Ireland take a turn.
The diagram shows my mic cut off. The diagram shows a pair of chromosomes, each of which has been duplicated in a neurospora cell preparing for meiosis. The chromosomes have genotypes A, B, and A. Uppercase A, lowercase b, and lowercase a, uppercase b, sure. Place genotype labels in the blank boxes to predict the genotypes of each of 4 daughter cells. Place only one label in each blank box. You may use each label more than once.
Oh, you're muted.
Oh, there we go. Showing us in this diagram chromosomes. And then what has happened here? What has happened here? Yeah. Good job, Ireland. That was great. They crossed over, right? It would be like if I was standing next to Ireland and I was like, hey girl, can we switch legs? And we chopped off one of my legs and I gave it to Ireland and Ireland chopped off one of her legs and gave it to me. How tall are you, Ireland? I'm 5'2", so we be walking crooked, all right? But that's kind of what we could do. I mean, if I could break off one of our girls' Barbie legs and swap them, that's kind of what is happening here, right? So let's try to do this. Let's draw on the board right now what we think. I'm going to give you privileges if you don't have them. My default permissions are weird.
Yeah, we have souls.
Perfect. So what do you think? We're going to start down here with this cell, right? The yellow cell, the yellow one is A, big A, little b. So then let's look down here. Is it the same or different? What goes in this box? These are what we're going to drag. So if you were taking the test, these would be what we can drag over. Somebody draw a line. What are we going to draw? What would this chromosome be? This first left yellow chromosome right here. Well, if I look in this diagram, it's big A, little b, and this is all yellow, right? Good. So big A, little b. All right, let's move to the next. All right, so all yellow chromosomes look like they're big A, little b, but this is blue. So I'm going to go over here and look for what the bottom blue part codes for, and it codes for B because that looks like what was swapped. So it would be good. Big A, big B. High five. What about over here? Yes. Little a, little b. All right. And then the full blue, little a, big B. How we feel about that? Green check, red X. That once we talked through that, I feel like you did a great job. And you did that with confidence. Good. Super easy, they said. All right, then we spend no more time on this one. Let's move on down to our next. Do I have a reader in the house? What do you think? We've got a two-part question. Go ahead and read it for us, Pip.
All right. Oh, it's these ones, whether you love them or hate them. Anyways, the following question has two parts. First, answer part A, then answer part B. Part A. Which statement describes crossing over as it occurs in meiosis? A. The alleles of a gene separate. B. Spindle fibers pull apart sister chromatids. C. Genes move to opposite ends of the chromosomes. Or D. Homologous chromosomes exchange segments of DNA. Could I read part B?
Yep.
Okay, part B. Which method do scientists use to confirm that crossing over occurs? A. Cloning. B. Dihybrid crosses. C. Gel electrophoresis. Or D. Genetic engineering. Thank you.
Good. All right. What is Mrs. Rittenhouse going to tell you about how we're going to do this question? What's going to be my first mode of recommendation? What are we doing first? What would I tell you? Test strategy. How are we going to attack this question? Read it carefully. Good. Oh, and identify keywords. Good. So I like that. So first of all, does this question give us any information? Sometimes it's nice when the questions give us, remember, actual content. Does the question give us content?
Oh, Ren.
Does the question give us any answer? The question is really lame, right? It doesn't give us any content information. So we're going to just cover up part B, right? Everybody cover your part B up. We're going to focus in, and you guys are already doing a great job of underlining keywords. So we're looking at what describes crossing over in meiosis. All right. Some of you said get rid of bogus choices. I love that. So let's get rid of them. When meiosis occurs, yes. Milo,
Oh, run.
what do you think? What are some of our bogus answers? The alleles of a gene separate.
Can I have a potato?
Yes, go sit down at the table. Spindle fibers pull again apart sister chromatids. Genes move to opposite ends. Homologous chromosomes exchange segments. All right, so Pip says let's get rid of A. The alleles of a gene separate. All right, this would actually be this one would actually be part of a phase. Does anybody know what happens when the alleles of a gene separate? What phase is that? Yes. Anaphase. You're absolutely right. That's anaphase, right? When they pull away. And that's not when crossing over occurs. It doesn't have anything to do with crossing over. So that's a good one to get rid of. Good job, guys. Okay, I like that. So exactly. The genes are moving to opposite ends of the chromosomes. You're like, that doesn't make sense either. Our genes aren't moving on the chromosomes, right? The genes aren't moving on the chromosomes. So that's good for my computer. So then we have spindle fibers pull apart sister chromatids, or we have homologous chromosomes exchange DNA. A lot of you are putting in chat now, C, 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 C. And I think you're right, right?
Actually, it was D, I think.
I mean D, I'm sorry. D, 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 D. Yes. And I would agree with you. All right. It is going to be D. Our homologous or similar chromosomes, they're going to swap DNA. That is what crossing over is. So then let's move this down here, this box. Now we're going to move it up to, now let's check out the second part. Which method do scientists use to confirm crossing over? Oof. What do scientists use to confirm? How do we know crossing over occurs? Can't be A. All right, I like this. We can't be A because cloning means what? A clone is what kind of a copy? A duplicate, an exact copy. So we can't confirm crossing over because crossing over gives us differentiation, right? Crossing over gives us variety. So get rid of that. Good. So are we going to use dihybrid crosses, electrophoresis, or genetic engineering? What do you think? Tell me more, Dustin, about why you think it'd be gel electrophoresis. LOL. Yes. So you're absolutely right. Gel electrophoresis is actually the answer. I think a lot of us are getting thrown off on this one, so I'm just going to share it with you. So what gel electrophoresis is, it is a plate. And what we do is we put a piece of DNA down here. All right. And what it does is it sorts out the DNA segments. And if you look at them, they're all different lengths. So if we took two reproductive cells and a somatic cell, meaning maybe one of your cheek cells, or let's just say one reproductive cell and one somatic cell, right? So I'm taking two of yours and I'm running a gel electrophoresis, you would probably see that there is some differentiation in the alleles of the chromosome because of the order of what's there. Okay. So it would be gel electrophoresis. Let's look here. Here's our DNA, right? In DNA, adenine always pairs with what? Thymine. Who remembers what apples grow on? Apples grow on trees. Good. And what about down here? Chips and cheese. Ren likes chips and cheese.
Chips and dip sounds legit.
What I like is guacamole. So chips, C and G paired together. Chips and guacamole. Okay. C and G. Honestly, as a kid, I always remembered C and G went together because they look so much the same, right? Like a G is seriously a C with an extra line. And if you can remember one, then you get the other, right? So let's go to a question on DNA and check our DNA knowledge here. Is this it? No. I'll skip ahead some of these and here we go. All right. Somebody want to read? Any readers in the house? All right, let's give Ireland another chance, and then Pip, you can be up again next.
In 1961, scientists used repeating RNA segments such as UGUG, everybody gets the idea, to build amino acid chains. Based on the amino acid chains built, they assembled the codon chart shown. A scientist investigates the effects of a single base substitution mutation in a 3-base DNA sequence. This 3-base DNA sequence codes for an amino acid. A table containing some of the scientists' data is shown. A. Fill in the original strand boxes in the table based on the amino acid produced. B. Fill in the mutated strand boxes in the table based on the DNA sequence.
Great job. That was a lot of reading. I want to tell you, last year, from sitting where I sat at the test desk, I saw these codon charts. I saw these amino acid charts. Look at it. I really want you to look at it and familiarize yourself with it. Guys, this codon chart is not something that you have to have memorized. You just have to know what it is. Amino acids make what? Please tell me in chat. Yes. Amino acids are like those beads. Remember those beads from the ribosome? We read the mRNA, which is a copy of the recipe that comes from the nucleus. RNA is a code of our DNA, and it goes through the ribosome, and the ribosome reads it, and as it does, it breaks it up into three letters at a time. 3 letters at a time. And all those 3 letters will code, which is why it's called a codon. They will code for a specific amino acid. And then at the end, there's a code that says cut. And it cuts it, and they spin and twist all upon each other, and they get a shape. And the shape of that protein equals its what? Yes, function. Shape equals function. So I don't want you to be overwhelmed when you see a question like this. Say this to yourself. I will see a question like this. It's like you get a peek into the test right now. Almost 100% guarantee you will see this. So let's see what we are going to do. All right. The first part says fill in the original strand boxes in the table based on the amino acid produced. Okay, so this is the amino acid produced. TRP. TRP. Well, let's look over here. The first letter is a T. So first position, right? Let's look for the amino acid in there. Sorry. TRP. Who can find it in the chart? Where's TRP in here? Yep. It's right here. All right, let's highlight that sucker yellow. Somebody want to highlight it yellow? That's what we got. TRP. So the first position. All right, it's going first position, second position, third position. All right, everybody pop off there for 1 minute. I'm going to move some of these, not because I just think we've got now so much on here we can't see it. I'm moving some of them. Here, I'm going to stick one of these back. Actually, like to that right there. Oh, TRP. Okay, I'm going to shrink it down. Perfect. TRP. That's where we're going for. So then we see on the chart where it says first position, second position, third position. So let's look at TRP. What would be the letter of its first position? What box? U. So let's write a U over here in the box. That is our first position letter, U. Aria, will you be our steno? Will you put that in there for me? A U right here in the first mRNA box? Because we're working backwards in this question, okay? So U. What is our second letter? The second position is up. So let's find TRP and go up. Its second position is, or second letter is G. Now let's look at the third letter. What is the third part of its codon? Here's the third position. So TRP over G. Good. So the mRNA sequence is UGG. Now, how are we going to find the DNA sequence? We know that in mRNA, there is no what? T. Good. There's no thymine. So what does a U is representing a T? So what pairs with T? What would be the first letter of our DNA sequence? What pairs with T? A. Good. So let's put A in there. Perfect. A. What about G? What pairs with G? G pairs with, yeah, C. G pairs with C and G pairs with C. So our DNA sequence would be A, C, C. How do you feel about that? We just did that first part of that question. That was perfect. Give me a smiley. We just worked backwards. We found the amino acid, and then we followed the chart, first position, second position, third position, to find the RNA. And then we went backward the next step by using what we know about the letters that pair together. Okay? Who has questions about it, or is it just overwhelming? I see some meh and some, there's a couple sad. Tell me how I can help reassure those sad and meh faces. Think about it and put it in chat or send it to me privately. Okay? Let's do the second part. All right? Fill in the mutated strand boxes in the table of the DNA sequence. At the end of the day, guys, I don't care if it's mutated. Our job is just going to be to take these and translate. All right? So TCC. TCC. Absolutely. Pop on mic, Aria.
Okay, maybe. Okay, there it goes. So for this column, I was like reading the UCAG parts, and then I realized that there are 4 letters here. And I was just wondering what the stop means and why there's 4, because we normally only have like 3 letters in a sequence.
Good. So there's 4 letters because even though there's only 3 in a sequence, there's 4 total possibilities. So you'll see for each position in this chart, each box in this chart, it gives you all 4 options, because even though there's only 3, yeah, there's a possible 4 that it could. But that's a good question. You're really thinking about it. I like that. And then a reason that there are stops, that stop means that those stops will come at the end of the amino acid. And that's actually the code to like stop and cut it. Like, this is the end. Does that make sense? It's actually the stop. So the ACC, we got this code because we answered this question. So how do we get the amino acid? Well, we went here and did U, G, G. So this is the copy of the DNA. So we know that if we have the RNA copy, that we can pair our letters together, and that will give us what the DNA copy is. Does that make sense? If you have a DNA copy, you can figure out the RNA. And if you have the RNA, you can figure out the amino acid. It's kind of like one, you have to have one to get and understand to the other, right? If they just gave you, so basically we are using these as steps to get from one to the other. So in this first part of the question, we worked backwards. So it's like we walked up the stairs, right? Down here, we're going to get the DNA. And now we're going to walk down the stairs to figure out what amino acid was produced. Does that make sense? Okay. I know we're running close on time. What I'm going to do is, if you need to leave. I want to run the end of this question for clarity and for the recording. If not, you can come back and watch the recording and get the answer to this. I'm going to show you the second part. It is TCC. All right. Usually T pairs with A. Apples grow on trees, right? So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So we are going to go ahead and do that. Apple grows on trees. So So T codes with A. What pairs with C? What pairs with C? Good. So if we have a C in our DNA combination, then as our RNA reads it, we're going to get a G. Is this not working? And if, again, there's another C, if the DNA reads C, the RNA is going to read G. So we get A, G, G for RNA. And then we are going to take that code, A, first position A. So here's first position, I find A, so I know we're going to be in here. The second position is G. So A, G. I'm going to match up those columns. Okay, we're in this quadrant. Now, which of these four is it going to be? Third position is G. So it's going to be ARG. A-R-G. Does that make sense to everyone? A-R-G. You are right. A and T match right here, Justin. A and T. Now this right here, and the mRNA, we're not matching. This is where our code. This is our code that we're taking to the chart. So our mRNA then we are taking to the chart and just matching it up. Does that make sense? Well, for it to, why not UGG? Because there's not thymine in mRNA. But the thymine is on the DNA side. So when it transfers to RNA, it still T still matches with A. Does that make sense? You still have adenine in our mRNA. You just don't have thymine. Yeah. Just no thymine in mRNA. So if you ever see thymine, you know you're dealing with a DNA sequence. So the DNA sequence, help me, Kylie, with what your question is. So in this case, they gave us the DNA sequence. That's what they give us. So we work from the DNA down. In this sequence right here, we're working backwards. So we're using that mRNA sequence. The guacamole pairs with chips. The guacamole pairs with chips. The uracil, which is in replacement of a T or a tree, pairs with an A. I don't know if that helps. I think that's where you're stuck maybe right here is finding this DNA sequence. We use
I think
the
it's Justin asking the question.
mRNA code. Nope, I had another one in private chat.
Oh, never mind.
The TRP went to UGG. These are just two separate questions. So I just want you to keep those two separate. Yep. If you're saying what is mutated, you're right, Justin. The mutated sequence is the A and T got switched. Nope. In mRNA, the G and C stay the same. Same thing with the A. So the A, G, and C, they're all the same. There's just, for some reason, no random thymine in mRNA. I can't tell you why. It'll be a mystery that we can ask a higher power someday. So there is no chart for the DNA and RNA. There's no chart for the DNA and RNA. The DNA and RNA you have to keep in your brain. All right. So this, if I had to make a chart for DNA and RNA, I would do this. And yes, I know it's late. So if you need to go to another class right now, go ahead and go. If you need to stay to understand something, stay. All right. This is DNA. In DNA, our adenine, our apple always pairs with our thymine. And our cytosine always pairs with our guanine. Apples grow on trees, chips, and guacamole. Okay. That's our DNA codons. That's how they always pair together. Now, they can be on opposite sides. They don't have to be on a certain side of the strand, but just T always matches with A always matches with T. Now, in RNA, okay, in RNA, it's almost the same. All right, it's almost the same. Our RNA chart would say that A pairs with U. So if there's an A in the DNA strand, it will pair with a U. There's no T in RNA, and there's a C that always pairs with G. So this is essentially the chart. You just they don't give you that chart, right? That's the chart you have to have in your head. You have to know those base pairs to answer this question. Does that make sense? Okay. So these are what we know. This is what the question wants you to know in your head, right? And they want you to know that you take this mRNA sequence to find the proper amino acid order. Hopefully that helps. I think we have another question like that we might peek at tomorrow. Okay. We're going to peek out another couple like that tomorrow to make sure we understand. So we'll come back to this tomorrow if you're still. Yep, absolutely. If you remember those four, you're golden. Apple grows on trees, chips, and guacamole. Yep. And then in RNA, there's just no T. So anytime you would go to write a T, just cross out that T and put a U. And C always pairs with G no matter what molecule it is. As long as you remember that, that's all you have to have in that noggin for these questions. All right, we're way late. I'm going to let you go. I'm going to end our recording. Have a great day. Pop into a help session. We're going to do more of this tomorrow, so don't feel overwhelmed, okay?